the youngest and the stellar scientist I know. So a little bit about Arjun's academic career. So he's, I mean, and scientific together. So he's a computational structural biologist at IIIT Delhi in our department. He earned his MPhil from KTH Stockholm, and after that he did his PhD from the CSIR IGIB from New Delhi. So his formal education is in the field of chemistry as well as biophysics. That is why he got enough interest in deciphering the complex biological processes by understanding their three-dimensional interactions. Now, a little bit more about what his lab actually does. So his lab is primarily developing methods and tools which, apart from helping the community to use his resources, but also enables his own lab people to understand the field of structure prediction, force distributed analysis of lipids, deciphering the mechanistic understanding of the interactions between various biomolecules, and they're working in a multi-scale systems. As a structural biologist, he has been regularly working with the techniques for structure prediction, molecular dynamic simulations, and biomolecular docking. So I leave the stage for Arjun. Please go ahead and share some of the exciting science.
Thank you so much, Gaurav. And first of all, thank you to Professor Chandra for an excellent talk. There are some overlapping things which I would love to discuss later on with her. And especially good morning to everyone on this. It's a beautiful Monday, rainy morning. So let me just share my screen. Gaurav, can you just once confirm if you can see my screen?
Yeah. Your voice is clear. Your screen is very clear. So please go
ahead. Okay, perfect. So today in the session of molecular modeling, I wanted to present this study which is ongoing in my lab, and it is the talk is going to move from actually an analytical tool, structural biology analytical tool, and we are going to explore its utility to finally result in molecular modeling of actually empty space. Till now, I think the talk by Professor Jairam was all about the actual sequence and the structures of protein. I'll be taking it a little step back, and I'll actually be talking about molecular modeling of empty spaces. And hopefully some of the insights people may enjoy, and we surely in the lab are excited over it. So to start with, I would like all of you to take a moment and look at this cup. Now, if the most simplest question all structural biologists generally ask is, what are the accessible residues on this protein? Now, if I asked you in this context is, what are the residues that constitute the inner lining? What are the residues that constitute the inner lining? It's actually seemingly very easy question, but it's actually a very difficult question when you consider that the current way of generally finding out accessible residues is that you roll a probe onto the surface and you find out which are the residues which are maximum in contact, and you call them as accessible. If you had to roll this probe onto this cup, you realize that everything inside as well as outside is accessible. But that would not answer this question is, what are the residues which are only part of the inner lining and not the outer lining? So from here, we started the question, and as I said, it's going to be 100% accessibility, both sides. So taking a step back, and we talk about protein cavities as the cavity of that cup. So cavities can broadly, and this is the only part where I'll be talking about a little bit of definitions to bring everyone up to page. The cavities can be of these three types, which are the channels which connect 2 points, and it could be a hollow channel from one end to the other end across a membrane. It could be tunnels, and tunnels connect a point from the surface of the protein to the inside. So it's more of tunneling through the membrane in most of the cases. And then you have pores. And if I say that pores are actually a subset of channels, it's not wrong of a definition. Then another part of cavities you encounter very frequently are pockets and clefts. And pockets and clefts are these depressions on the surface of the protein. Pockets are generally shallow and clefts are a little deeper depressions. And finally, you have another classification, which are voids. So you can have, this is actually a virus encapsulation, and you can have it completely cut off from all sides, though there is obviously accessibility from all ends, but it's not obvious which is the connecting points between these. With this definition, most of you already surely know about this, but protein cavities are very, very important protein function classes. You have, especially when it comes across membrane activities, you require something which will transport something from inside to outside or vice versa, and proteins cavities actually do that. It could be actually a linear transport, or you can have an alternate where something goes in and then the protein undergoes a conformational change and it releases it. Also, it could be a site for catalytic activities, as well as these cavities could be part of, we'll come back to this ATP synthase molecule especially, where this cavity actually takes part in rapid transition between different states, and this cavity acts like a groove. So when we started with this project and tried to answer the simple question, which are the residues part of inner lining, these were generally the tools which were present. And you'll see that on one of the criteria, which is the atomistic detail, so which are the atoms which are inside. Only one method used to give this. You had residue level information from some of them. None of the methods actually gave you an understanding of what is the volume of the cavity. Similarly, hydrophobicity scale, downloadable, you required especially a lot of human intervention in all of these methods, which means is you would need to pinpoint what you define as a cavity. And based on that, the program would find out which are the residues which are part of this cavity. So with this, we actually developed a method called Cyclop, which pretty much covered all the lacunas. In fact, this is a little old. Now we also have a standalone program which you can download and run it if it requires on multiple structures. So all of the lacunas that were there in this field, we kind of tried to address that. And in the most simplest way, if I say that what the method does is that it takes residue, your crystal structure, or it can be a model structure. And it doesn't matter how you save the alignment of the structure, the Cyclop will actually automatically rotate the structure to find out the maximum, maximizing the z-axis for, and then it will create a voxel grid and find out what are the inner residues. And this is the web server for that. But a simplified version of this algorithm is that first we actually find out what are outside, what are inside, and then we try to find out which is the middle ground, and we take off the outside. So what is left is actually sampling of only the inside atoms. And since we do it on a voxel grid, so we actually get atomistic detail information. And sorry, I should have put this forward. Uh, which means is now it's up to the user to define if I want something which are 100% accessible versus 50% accessible versus anything less than 50, you don't even consider them. So now I can have a gradient, and it's up to the user to say that of a residue, let's say arginine, lysine, whatever, how many percentage of atoms should be accessible to consider their residue to be 100% accessible. Anyway, so that is an atomistic handle that becomes very useful. The files that you actually get out from the Cyclop output is you have all the residue listing as a residue DAT file. You have the inner surface mark as a B factor. You have a PQR, which actually maps the cavity, and it also calculates the conservation scores, gradient profile. I'll go through some of the case studies to show the utility of all these parts. And this is what the summary file which you get. It calculates all the number of residues, more than 50%, which are not completely inside. It will give all the secondary structures on which these residues are, and it will calculate all the hydrophobic, hydrophilicity, and it will also tell you if the overall cavity is hydrophilic or hydrophobic. And this is just to show that this is how it would mark the B factor. So if you have residues which are part of the inner surface, then it would just mark it as a distinct unique number, and rest would be 0. So you can utilize that to color your structures. So if you color the structures, we actually tried it out on several different shapes of cavities, and this actually introduced us to the wonderful world of cavities, where you have even shapes like these forks. You have something which is like a zigzag pattern. You have shapes which is almost like a cavity which shows as a cross. So there are various ones. And then when we took the method for further validation, we thought that the accessibility, when we are interested, what it means is accessibility to water, right? So the easiest way was to use water dynamics and have a very short simulation where the structure actually does not alter. So it's less than 2 angstrom of deviation. And we took a structure and we just wanted to saturate the contacts of the residues with all possible water combinations. And if we saw that a residue is at least 80% in contact with water, we considered that as accessible. And from there, when we tried all the other methods, you can clearly see that Cyclops supremely overperforms in terms of precision at 90%, 91%, and accuracy at 91.5%. That gave us even further confidence in our method, and we took quite a diverse set of structures. And if you look at representation, so this panel, A panel, is just isolating the cavity and the residues. And you can see that most of the methods, like here in Mold Online, Core Walker, structure like these, they completely miss out on what is the inner residues. And these are inner residues which they mark. So when you talk about super structures and super macro structures, we see that the sensitivity of all these methods actually diminishes. And Cyclops has been, at least qualitatively here, also been able to outperform its previous legacy. So let me share a couple of case studies where studying such cavities through Cyclops can be useful. So I told you about a minute ago about ADP synthase. So ADP synthase is a very interesting motor protein complex where it actually undergoes a 7-stage mechanism. and I won't go into the biology of it, but this is the first time actually using Cyclops. Now we could actually detect how in every state, because there was a triome structure of all the 7 states available, we could now characterize for the first time how the volume of these gamma subunit and the cavity changes. And similarly, we could also find out how the change in the spore volume creates new residues to come towards the surface or make them buried. And the hydrophobicity, interestingly, residues are constantly changing. Also, it is well known that ADP synthesis actually, the mechanism actually has a piston kind of mode of action. And Cyclops was able to capture this. So if you look at this, and this blue line is the gamma subunit, if you look at this blue line, you can see that in state 1, state 2, and state 3, imagine this is actually the gamma subunit, which is coming from below, and it is in a rest state here. It pulls back here and extends here. So this is the mode of action that actually the ATP synthase people know of. But now with Cyclops, we can actually calculate and find out which exact residue and atomistic scale does this perform in. Another case study, and this is another study that we performed in our lab. So this is a huge protein molecule which is known as ABCA1. It's a very critical protein when it comes to cardiovascular diseases. It's a 2200 amino acid membrane protein. And we wanted to study this ABCA1 and triome structure. And there were several hypotheses drawn based on the triome structure, which we wanted to evaluate in its native lipid environment. When we took this structure and analyzed it with Cyclops, first of all, we could see that we had perfect agreement of hydrophobic region, which is anyone would agree with this. With a lipid environment, you had hydrophobic residues all around. You had a region which was vacant of actually charges, which was quite surprising. And as I said, Cyclops calculates the conservation scores also. So with this conservation score, we actually found 3 residues which were part of the inner lining, highly conserved, and any mutation to these are reported in literature to cause severe pathogenic diseases. So perhaps though we have not evaluated the mechanism, perhaps the fact that these residues line up the core of the inner cavity, and perhaps these residues have a key role to play when it comes to lipid transport, that it is functionally important to be highly conserved. And this fact that we could find out the residue's functional site will give more critical insights of why those diseases occur. One theory that was proposed in that triome paper was that they said that there was a section of the structure which was not wide enough to accommodate any lipids, and henceforth they proposed an alternative hypothesis. And we actually showed for the first time is that if we simulate the structure in its native heterogeneous membrane and taking care of all the parameters, that actually the point where they're talking about being very narrow, the volume is actually not that narrow. And in fact, it is wide enough to accommodate lipids. This paper is under review and it's under revision actually. So hopefully we would be able to contest the findings in the crystal structure. Yes, so this is about the ABCA1, as I said. And so moving on, another key application we actually identified using Cyclops is This is a work in collaboration with Kaushik Chakraborty in CSR IGIB, whose lab's interest is on royal chaperone proteins and how proteins are folded in those chaperones. So with Cyclop, we could actually identify all the residues which are 100% inside and not with this identification, what we could find out was that all the residues for all the organisms, especially on low order, what were the electrostatic charges? And when we considered, when we have these residue informations, we could actually utilize this to see what is the central or the centroid charge of these cavities for the first time, because experimentally it's not possible to calculate what is going to be at the centroid, what is the charge, electrostatic charge. But now, since we had all these residues, we could extract what are the electrostatic values of every single residue, and henceforth, what is going to be the summation of the centroid charge. And when we map that, we interestingly find that the cis and trans cavity mimic each other, which is the top and the bottom cavities, and also how the charge evolution happens in these cavities. More interestingly was that if we look all the way from human to yeast, there seems to be an evolution of how the chaperone lining itself has evolved. So it seems that with the human proteome, the chaperone has turned positive, while when you talk about lower order, Goyel, so E. coli, yeast, and everything, it is more towards an highly negative. You have cases which is this HHA, which is almost close to minus 54 in terms of electrostatic. So this got us thinking of the role of electrostatics in terms of these evolution. And what his lab did was very interestingly, they created chimeric chaperones. So they took actually something like CFL and Goyel or a yeast with another SMH or with human, and they started creating constructs where they would only mutate key residues at the interface where we could identify which are the inner lining key residues and highly conserved. So when they changed that, we could actually modulate how the cavity now electrostatics values were. But this was interesting as a fun experiment. What was more interesting is the data that I'm not showing here is that with changing the electrostatics, we could change the folding rate of protein inside a chaperone. So it seems like more electronegative chaperones folded faster the same protein or an ortholog of it, while human proteins actually had slower kinetics to fold the proteins. Perhaps it could be that nature would want the chaperone to take its time and do it correct. So we could actually play around then with all these kinetics of protein folding within the chaperone. But again, this is an example where knowing the inside lining of a cavity, one can decipher the function of proteins. So with that, we actually moved ahead and asked this simple question, especially since we were talking already about evolution of electrostatics, is how did protein cavities actually evolve? So what I'm trying to perhaps say is that was there that the lowest form had a square which turned into like shape like this, then which had an edge like this, and finally, perhaps in humans, we have evolved a shape like this. So are there a shape evolution for the same function across the species? Since we had this handle of a tool, now we could actually study these shapes. So the first question was, what is nature's encyclopedia of protein cavity shapes? And second was, do cavity shapes evolve over time for the same function? And thirdly, are there specific shapes associated with specific activities? And this is where we are now entering the domain of molecular modeling. So Atul, in my lab, he started studying is deciphering the underlying evolutionary context of protein cavity conformations. So first step was to take out all the protein cavity bond structures from the PDB, and we found that there was a wide range, which was all the way from 300 to 500 to up to 5,000 plus structures were present. And we did an evolutionary mapping of it. But we kept the evolutionary mapping for later on. And first thing, our challenge was, now that we have these inner cavities, which were actually voids, if you remember the previous images. So now we had to map and create these images and characterize them, which means is that we had to have a correspondence to understand, is this similar to this or is this similar to this? Which is not an easy thing when you think about shapes. So we took majorly three shape descriptors, which was eigen-based in heat kernel signatures and wave kernel signatures. I'm not going to go into the details or the mathematics of it. I can answer that in case anyone has specifics later on. So first thing we try to do is, in terms of eigenvalues, heat kernel and wave kernel, was classify all the proteins from different function base. So I'm just showing one example, which is enzyme function, which is enzymatic proteins. And what we found is that eigenvalue actually could discriminate between the different similarities. And this is what I meant by eigenvalues. So we could actually discriminate much better the whole population of enzymes, while heat kernel and wave kernel did not exactly function that well. Also we saw that there was dissimilarity among shapes, and we classified from the very onset, we actually changed the module from single chain and polychain to be treated as separate, because I'll just come in a few slides to show you what is the importance of cavities which are formed by single chain structures, while some cavities which are formed with multiple chains complexes. Also, we developed a whole algorithm, and you can see that these are some cases where we identified similar structures, and you can see the similarity. So even though the size of the protein, let's say 1H1 and 2AOAR, the size of the protein inherently would make you say that they are not similar. But if you had an algorithm which could map out the exact points, you would see that they perfectly overlap with each other, saying that at least there is some similarity in the shape compared to others. So these are just three examples of that algorithm that we developed. Then we came to the first question which we had was, does nature actually have an encyclopedia of shapes? So this is, again, just I'm showing one example of channel proteins, and what we could find was, so this is single and these are polychain, and we could find was, in eigenvalue there's a wide distribution of shapes, and the shapes actually range. In terms of statistics, there is actually a range in those shapes, how they look like. And though these shapes may seem very similar, quantitatively, if you look at it, there is actually quite difference between these two shapes. Then we moved ahead and we started performing fractal analysis. Now, fractal analysis, for the people who may not know, is a very interesting thing where we talk about how disordered or how broken is the surface. So ideally, a piece of paper, a plain sheet of paper, would have the least fractal index, while a completely crumbled paper would have the maximum fractal index. So if you look at this, so you have something like here, which is the least fractal index, where it's kind of very smooth, while you have high fractal index when it's completely you have depressions, contours, and a lot of disorderedness on the surface. But remember that this is actually the inside surface. So this is kind of the negative image of the cavity. And one thing which immediately pops out is that the single chain actually has a wide range of cavity shape fractality, while the polychain seems to have a normalized distribution to it. And this is something that we observe in multiple cases when we talk about binding proteins, when we talk about channel proteins. We constantly see that there is a wide range in terms of single chain versus there is a continuous normalized distribution in terms of polychains. And you can see that the distribution actually is, you have safe surfaces like this, while you have extremely disordered and broken surfaces like this one, which is here. So again, transporters show the similar thing, and this is a class where we could not classify exactly the protein into a functional form, so we have kept them as separate, and this requires more in-depth study into it. Now, when we took a look at the density function of these fractal dimensions, we can automatically see that there is, first of all, there seems to be a bimodalness apart from toxin, because I would disregard toxin because there was very little representative structures in this class. But there seems to be a bimodal nature of the cavity shapes in terms of fractal dimensions. While when we talk about polychain or complexes, you seem to have all of them falling into a very mean median fractality. Now, this poses an interesting question, which again, I yet am not able to answer, but is it that nature has already optimized the fractality or the disorderedness of the complex structures or polychains, that everything falls in this region, while these single chain ones, because they have the flexibility of folding and required to do multiple individual functions, that there seems to be still a lot of processes which have different requirements of different fractality. We don't know. And this is, again, an open question for us. With this, I would talk about my ongoing efforts in these dimensions. And we are actually now mapping these structures and insights into the evolutionary tree, and hopefully try to answer the first slide that I showed, where it goes from rectangle all the way to something else shape. Does it actually follow a pattern? Since we now can quantify every step of the shape. We will also be having a GROMACS version of Cyclop very soon, so one can actually look at evolution of the inner cavity over a simulation time. And an interesting implementation of Cyclop we are trying to perform is where we study how the water dynamics inside cavity shapes change, and if water dynamics has a role into, especially in terms of chaperones. We surely know that water dynamics will have a role to play because of the electrostatics. So these are the three implementations of Cyclop that we are still working on. And with this, I would like to acknowledge, obviously, my students who are the backbone of all these works: Parth, Sukriti, Mrinal, Pratyatul, who developed Cyclop, Sunidhi for the ABCA1 study, Triple ITD for basically providing these amazing students, as well as the facility and all my collaborators. With this, I will stop, and in case anyone has any questions, I'd be happy to answer.
So thank